morning, everybody. Today is 20, October 1st of 2024, and we will have OSEAN's weekly seminar and international seminar. We will study the paper titled The Potential Impact of Nano and Microplastics on Human Health, Understanding Human Health Risk. Alicia will present the abstract of this seminar. Alicia, the floor is yours.
Hello, everyone. Good morning. First of all, there's a lot of medical terms in this paper that I've never heard before. So most likely I'll pronounce it all wrongly. So sorry. And secondly, today is actually public holiday in Korea. So my brain is a bit fuzzy. Okay, so moving on to the abstract. This is the abstract of the paper. Plastics are used all over the world. Unfortunately, due to limited biodegradation, plastics cause a significant level of environmental pollution. The smallest recognized to date are termed nanoplastics and microplastics. These nano and microplastics can enter the human body through respiratory system, their inhalation, the digestive tract, their consumption of contaminated food and water, or penetration through the skin via cosmetics and close contact. Bioaccumulation of plastics in the human body can potentially lead to a range of health issues, including respiratory disorders like lung cancer, asthma, and hypersensitivity pneumonitis, neurological symptoms such as fatigue and dizziness, inflammatory bowel disease, and even disturbances in gut biota. Most studies to date have confirmed that nano and microplastics can induce apoptosis in cells and have genotoxic and cytotoxic effects. Understanding the cellular and molecular mechanisms of plastics' action may help extrapolate the risk to human. So this article provides a comprehensive review of articles in databases regarding the impact of nano and microplastics on human health. The review includes retrospective studies and case studies of people exposed to nanoplastics and microplastics. So the reason why I chose this paper was because, well, it is common knowledge that plastics has been found in the human body. We don't exactly know what these effects may do to our body. So I thought it would be great that we read and discuss this paper to see what is the current knowledge known. So starting with the introduction, this paper starts with giving us background information about plastics, such as plastic production as shown in figure 1, and polymer types as shown in table 1. Then they provide descriptions of nano and microplastics, current status of global plastic pollution, and the current research on plastic ingestion and inhalation. These are some of the highlights of the introduction section that is typically typically, plastics are categorized based on their chemical composition of their primary polymer and any side chains, which includes acrylics, polyesters, silicones, polyethylene, polypropylene, polyurethane, and halogenetic plastics. The biodegradability of plastics is influenced by the raw materials used in their creation, their final chemical composition and structure, and the conditions under which they are expected to degrade. As of now, research is mainly focused on quantifying human inhalation, ingestion, and dermal exposures while considering factors like exposure frequency, duration, plastic size, or intake rates. However, different environments, spaces, and diverse population groups have varying exposure levels, just as we previously saw in our July seminar on the ingestion of daily microplastic across 109 countries by Zhao and Yu. So the primary concern for this rapidly evolving field includes potential for physical damage, toxicity, and inflammatory responses to plastic in the human bodies. In order to address that concern, this review discusses the biological impacts of nano and microplastic on human health. However, given the current limited understanding of nano and microplastics' effect on human health, the researcher of this paper aimed to discern if in vivo study can shed some light on the risk tied to human exposure. The method that they used was searching through PubMed for keywords such as nanoplastic, microplastic, human cell lines, human digestive tract, human intestine, human respiratory system, human lungs, human skin, and human nervous system. They also use term combinations such as two search terms and multiple search terms using logical operators OR and AND. In the end, they assessed 123 titles and abstracts for inclusion in this review, which included retrospective studies and case reports of people exposed to nanoplastics and microplastics, and grouped them into three sections. First is the route of exposition and uptake of nano and microplastics. Second is potential in vivo effects of plastic exposure. And third is the effects of cell exposure to plastics in vitro. So first we will look at the route of exposition and uptake of nano and microplastics. Microplastics and nanoplastics can enter the human body through three primary routes. First in ingestion, second is inhalation, and third is potentially direct skin contact. The first route is through the gastrointestinal tract, which is believed to be the major entry point for nanoplastics in the human body. Ingestion primarily stems from consuming contaminated seafood, milk, beer, honey, sugar, salt, and bottled water. In one research, they found that 81% of 159 global tap water samples contained microplastics, which was mainly fibers. Other studies supported this with results of 5 to over 1,350 micrometers of plastic particles were found in mineral water. Interestingly, one article showed that particle count in both glass bottle and PET bottled water were similar, with counts reaching up to 6,292 particles per liter. These results are also supported by a case study which detected 9 plastic types in human feces, with PP and PET being predominant. The second route is through the lungs. Respiratory epithelium is a primary target for inhaled nano-sized plastics whose biological reactivity depends on their physicochemical properties. Airborne nanoplastics and microplastic sources include clothing, dried sludge, agricultural residues, and tire rubber dispersed due to their mechanical wear during driving. Autopsies on human lung tissue have evidence of the presence of microplastics, namely that polymer particles and fibers were detected in 13 out of 30 lung samples. The third route is through the skin. The skin serves as a protective barrier against various external particles such as allergens, but also acts as a site for topical drug application due to its accessibility. Given its immunological significance, the skin could potentially be permeable to nano and microplastics, but data still remains inconclusive. Only a few studies have reported low penetration of textile nanoplastics through the skin barrier, but it is unlikely that nanoplastics contained in polluted water can penetrate the stratum corneum. However, these particles might infiltrate the body via sweat glands, hair follicles, or skin injuries. Currently, microplastics of varying colors and sizes, ranging from more than equals to 800 nanometers to 5 millimeters, have been detected in numerous human samples, including lungs, breast milk, liver, spleen, placenta, blood, sputum, colon, saliva, feces, urine, testes, and semen. Two examples mentioned in this paper with confirmed detected presence of nanoplastic and microplastic in the human reproductive systems is a study examining 6 placentas. In that study, 12 microplastic fragments ranging from 5 to 10 micrometers in size were found in 4 of them, with 5 located on their fetal side, 4 on the maternal side, and 3 on the amniotic membrane. Another study on ex vivo human placental perfusion, the model, assessed whether PS nanoplastics could cross the placenta barrier and whether this process was influenced by particle size. The results show that PS nanoplastics up to 214 nanometers in diameter could cross the placenta barrier without affecting cell viability. This paper doesn't really go deep into the literature on plastics found in the human body, but if you are curious, you can refer to Laporta et al. 2023 for more information, or we can have a look at it later because I have the paper as well. Now, the next section is on the potential of in vivo effects of plastics exposure. Certain plastics can pose health risks due to their inherent chemical composition or ability to absorb and transport other harmful substances. So this section will delve into specific documented instances where plastic exposure resulted in diseases as shown here in Table 2. Looking at Table 2, we can see that these are the health impacts of nano and microplastic exposure based on organ-specific effects. The first column is the organ or system affected, the second is the potential diseases, and third are notes from the authors that we should keep in mind. We will go through them one by one, but before we go in depth about the contents of Table 2, it should be noted that it is currently unknown whether the diseases associated with plastic exposure are caused by nano and microplastic particles, so the exact mechanisms of how plastic reacts and whether nano or microplastic can actually trigger reactions to the human body remain uncertain. We start with the skin. There are limited publications concerning the effects of nano and microplastics on human skin. Most studies predominantly focus on potential allergic reactions. One case study showed an early account of occupational allergy linked to plastic gloves dating back to 1985, where allergic reactions were relatively rare and only manifesting in 5 out of 542 patients examined. This posed the question if the allergy was due to the additives such as BPA or plastics themselves. It was concluded that while plastics themselves can induce allergic responses, additives used in manufacturing plastic items are also a potential allergen. Next is gastrointestinal tract, which has 4 main study focuses. The first focus is that there's a positive correlation between the concentration of fecal microplastics and the severity of inflammatory bowel disease. Specifically, research on IBD patients revealed a significant accumulation of microplastics in the ulcerative lesion of the rectal mucosa. The second focus is on carcinogenesis, namely cancer. Report findings on environmental factors influencing the onset of different cancers suggest an elevated risk of colorectal cancer among individuals working in various sectors, including plastic and rubber industry. There is also a noted increase in pancreatic cancer mortality among workers involved in producing reinforced plastics. The third is microbiota. While there is a lack of human studies on microbiota, in vivo experiments on mice showed disturbances in microbiota composition. This is due to the presence of PVC microplastics, which linked weight loss to the accumulation of microplastics and chemical additives in the tissues. Exposure to PVC in adult mice also led to significant gut barrier dysfunction, microbiota imbalance, metabolic disorders, inflammation, endocrine disruption, and lipid and energy metabolism alterations. Lastly is food allergies, which is connected to microbiota. In short, studies show that plastic have the potential to alter how food allergens are digested, heighten the permeability of the intestines, and lead to intestinal imbalances, all of which could enhance sensitivity to food allergens. The next is respiratory system, which has a potential for lung cancer, OHP, obstructive bronchiolitis, and respiratory issues. First is on the toxicity of styrene. One study by the Harvard Center for Risk Analysis carried out in 1999 showed results of occupational exposure to styrene varied but generally remained below 10 parts per million, with higher concentrations of over 20 ppm found in industries dealing with fiber-reinforced polymer composites and deemed it safe for general occupational exposure. But other studies on styrene exposure in rodents confirm carcinogenic effects, nervous system disorders, respiratory toxicity, and significant increase in lung cancer. However, of course, studies on rodents did not conclusively link styrene toxicity to human health issues. Second is OHP, which is from occupational inhalation of antigens leading to an interstitial lung condition. In 2011, a review of available literature evaluated the association between styrene exposure and the onset of non-malignant lung disorders. From the cases discussed, bronchial asthma was diagnosed in 8 patients, while severe obstructive bronchiolitis was detected in 10 individuals. In 75% of asthma cases listed in that review, positive challenge tests were reported following inhalation exposure to styrene. Third is obstructive bronchiolitis, where a series of cases have been documented in workers at companies producing glass fiber reinforced plastics. All 6 patients diagnosed with obstructive bronchitis were involved in producing or processing glass fibers using styrene resins. Other respiratory issues include a study of Turkish plant workers where there was a 3.6-fold increase risk of respiratory issues in employees exposed to PP compared to the control group. The reported respiratory symptoms include coughing, dyspnea, and wheezing. In another study, the respiratory health of workers in a nylon flocking plant over a decade showed a correlation between the severity and progression of nylon flock-associated interstitial lung disease and a low baseline of diffusion lung capacity for carbon monoxide. Additionally, results of prenatal exposure to BPA was associated with a higher risk of allergic diseases, especially in female infants, and there was a correlation with low birth weight, increased risk of asthma symptoms, wheezing, and respiratory tract infections post-delivery. Next is blood neoplasms, which can induce leukemia and blood cancer. In a cohort study examining cancer mortality among 5,201 workers at 2 boat building facilities, researchers compared American boat builders with Danish boat builders. The results showed that while there was no rise in mortality from hematopoietic neoplasms compared to the general population, a significant association was found between the duration and the intensity of styrene exposure and the occurrence of neoplasms. For the American boat builders, a reanalysis was carried out in 2020 to reinvestigate the relationship between styrene exposure and cancer mortality. This cohort had initially occupational exposure to styrene from 1959 to 1978. A statistically significant association was found between styrene exposure and the onset of leukemia and bladder cancer. Working life exposure to 0.05 ppm of styrene was linked to an additional death of blood cancer per 10,000 workers. In Denmark, a study was conducted with 73,039 employees across 456 small to medium-sized enterprises engaged in reinforced plastic production from 1968 to 2011. Among the study participants, a total of 665 cases of 21 different blood cancers, each with a minimum of 20 cases, were identified. When adjusting for exposure duration, the risk of acute myeloid leukemia was found to be twice as high for those with significant exposure in the previous 15 to 29 years. For neurological effects, there are a few potential diseases of neurotoxic effects. Encephalopathy, unspecified dementia, and degenerative neurological disorders. In 2011, a study was conducted on the residents of US Gulf Coast, but this region is believed to account for over half of the US's total styrene production. The study determined level of styrene exposure by analyzing environmental styrene concentration and measuring the styrene accumulation in the blood of 874 participants. Data on neurological symptoms, including headache, fatigue, dizziness, numbness, visual disturbances, and nausea were collected. Results show that 1/3 of the participants reported experiencing at least one symptom. There was also a significant correlation between experiencing one or more neurological symptoms and being in the top quartile for estimated environmental styrene exposure. This evidence suggests that exposure to environmental styrene might lead to neurotoxic effects. For the Danish National Registries, a study was carried out with the aim of investigating the risk of encephalopathy and unspecified dementia in individuals with documented styrene exposure. Similar to the results from the US Gulf Coast, 228 cases of encephalopathy and 565 cases of unspecified dementia were identified in their results. Next is osteolysis or bone degeneration around bone prosthesis in a significant challenge in arthroplasty and is frequently observed as a complication following cementless total hip replacement. Most hip prostheses currently used consist of a metal or PE ceramic core, but evidence suggests that PE particles can contribute to hip osteolysis, and the quantity of PE particles shed from impact surfaces correlates with the severity of osteolysis. With an anticipated increase in life expectancy, the number of arthroplasty procedures is expected to rise, as is the risk of periprosthetic osteolysis due to elevated PE abrasion. For diseases towards the reproductive system, there is a noted potential for decreased fertility, gonadal damage, and reduced offspring weight. However, there are currently no studies describing the impact of microplastic and nanoplastics on human fertility and reproduction. The potential diseases to the reproductive system are actually results from studies done in vivo on animals such as nematode or roundworms, zebrafish, and rats. Some of the results of these studies include nanoplastics being detected in mammalian offsprings, indicating they might cross the placental barrier and accumulate in fetal tissues. For example, in another study where pregnant rats were intrathecally administered with PS nanoplastics resulted in a significant reduction in the weight of both the fetus and the placenta, which can only hint towards its effects towards humans. For genotoxicity, the highest exposure to styrene occurred in manufacturing of reinforced plastic products, where it is primarily metabolized in the liver. In one case study to assess the potential genotoxicity of occupational exposure to styrene, a study group of 52 employees from 5 companies producing reinforced plastic items and a control group of 54 workers not exposed to styrene and primarily performing office work was studied. The results showed that the study group was exposed to air concentration of styrene exceeding the time weight average limit value of 20 ppm and the biological exposure index, leading to a significant increase in SCE frequency and DNA damage. The observation of increased microsomal epoxide hydrolase activity in styrene-exposed workers correlated with a higher incidence of SCE, suggesting a potential link between exposure and genotype toxicity. Finally, we move on to the effects of cell exposure to plastics in vitro. Research on human cells has revealed that nano and microplastics can trigger various cellular responses, including cytotoxicity, genotoxicity, inflammation, apoptosis, and oxidative stress based on stresses such as charge of plastic, size of plastic, time of culture, concentration of plastic, and type of plastic as shown here in figure 2. These results were generated from the literature review of about 34. Sorry. What's going on? Sorry, hold on. These results were generated from the literature review of about 34 literature review listed in table 3 here. This table was really long, so it was difficult to take a screenshot, but that's okay because most of it are medical terms, cell sizes, and the results of each of these studies. Fortunately for us, the researchers of this paper provided us with a summary of this table, which is here. Among the plastic polymers studied, PS is most frequently used in research. Interestingly, PS does not rank within the top 5 polymers produced globally. So there is a gap in data regarding other plastic types, and particularly PET and PVC. The plastic particle size ranges from nano to micro and even millimeters, and some have different surface modifications which are now seen as crucial for the cytotoxic and inflammation effects. Determining the duration of exposure is also challenging, and it is unclear if the observed effects are acute or chronic. This is because there is a lack of data representing long-term exposure, even though plastic bioaccumulation in the human body persists for a long time. Another problem is distinguishing and extracting plastic particles from environmental and biological samples, as this impedes accurate dosage determination in studies, and there is no data exploring the cellular effects of extremely low plastic concentrations. Upon entering the human body, nanoplastics interact with many biomolecules such as protein, lipids, and sugars. And when these particles come into contact with biological fluids like serum or plasma, it is hypothesized that nanoplastics change their original properties and acquire new biological properties. Also, there are currently no personalized in vitro studies assessing cellular exposure to plastics, and as a result, the impact of protein corona development on potential toxic effects remain uncertain. In conclusion, current literature does not conclusively highlight the detrimental effects of plastics on human health, nor does it confirm its safety. Research on human cells has demonstrated that nano and microplastics can lead to cytotoxicity, genotoxicity, inflammation, apoptosis, and oxidative stress, among other concerns. However, in vitro studies have certain limitations, such as the duration of nanoplastic exposure and the accurate determination of plastic particle size and their concentrations. A multifaceted approach is essential in advancing research and methodology related to nano and microplastics. First, it is crucial to develop standardized methods for collecting and analyzing human samples, which include identifying and quantifying microplastic fibers in human tissues, like the lungs, and accurately measuring environmental plastic concentrations. Second, studying the impact of plastics on cells at very low concentrations is essential, as there is a lack of current data in this area. Finally, in assessing the impact of human health, it is necessary to develop in vivo testing methods to measure the concentration of different types of plastics in the human body, thus establishing a direct link between plastic exposure and potential health disorders. So that's all the information of this paper. Here are some additional readings if you are curious to learn more about plastics in the human body and its effects. And here are my takeaways and some open discussion questions. Thank you.
Thank you, Ellie. It's kind of difficult paper. And if you have any questions and opinions, please tell us freely. This is discussion time.
Hi, everyone. Thank you for the presentation. This is Lincoln from Tangaroa Blue Foundation. I thought that was a very interesting paper and presentation. I think this is becoming more common to discuss, even in the literature. I attended a seminar by the Minderoo Foundation on their umbrella review of the chemicals that are in plastics and their impacts on human health. And they found some very interesting and scary results. And I think as we find more and more plastics, like they're looking in the tissues, in the cells, in our human body, they'll start to link to the chemicals as well. So I think that we are only going to be seeing more of this as we move forward.
Yep, I agree.
Thanks.
Okay, thank you for your opinion, Lincoln. Yeah, we don't have any questions and opinions. It's kind of difficult, and just we can understand their result briefly. And there is no certain conclusion for our human health from microplastics and the nanoplastics. That's the point, I think.
Nothing's for sure, but it's just my concern, because this paper is saying a lot of things. Microplastic will eaten by water and food. I'm just worrying that the low-income person will gonna be more effective in this problem, so it can cause another inequality. So yeah, I just want to share this concern with the seminar. Thank you.
Very good point. Very
Thank you,
good point.
Min-sung.
I think I agree with Min-sung because most of these results come from US or Europe. So I think the results will be different from the results if carried out in Asia side, especially Southeast Asia side, low income. And based on the paper that we discussed in July, we saw that China and Malaysia was like the top for inhalation and ingestion. So I will be curious to see the results from China and Malaysia.
Yeah, that's good point. Okay, then we'll wrap up the seminar so early this morning. And
Okay.
thank you for coming. Bye bye.
For listening.